Today we have Anne Lightbody with us. Anne is an associate professor at the Earth Sciences Department at UNH. She's also an affiliate associate professor with Civil Engineering and Integrated Applied Math Department. Anne's research focuses on flow and transport within surface water systems, including rivers, streams, lakes, water, wetlands, and saltwater marshes. And today it's going to talk about some Fluvial reach scale solute transport in river systems. Over to you.
Thank you, Sashan, and thank you for the introduction. So I'm going to talk today about rivers. And I know that this is an ocean seminar series, so I wanted to start by giving you a reason why you should care, which is if you are interested in oceans, then rivers deliver freshwater, sediment, and nutrients and pollutants to oceans, of course, coastal oceans in particular. And if we look at estimates of how much comes in, we measure it in the teragrams. For example, for nitrogen, teragrams of nitrogen. This is an old study, but you can see that estimates sort of agree. Depending on exactly how you run the numbers, you get slightly different ones, but we're getting 10, 15 teragrams of nitrogen per continent per year. If we look over time, this is fuzzy, but the light blue is showing pre-industrial revolution in 1860. The medium blue is showing 1990, and then the dark blue is showing future projections in 2050. Again, not new, but this is showing that nitrogen exports have been increasing over time. And this is something that our oceans have to deal with. So rivers, again, they're delivering freshwater, nutrients, pollutants, and sediments to the coast, but they're not doing it, they're not delivering everything that they receive. For example, if we look at the Gulf of Mexico, we have a hypoxic zone, dead zone in the Gulf of Mexico that comes about as a result of nitrogen delivered by the Mississippi River. The bulk of that comes from fertilizer application to the Mississippi watershed. But not all of the fertilizer and the nitrogen that is applied to the watershed makes it all the way to the mouth of the Mississippi and enters the Gulf of Mexico. Only the portion that enters right close to the exit actually makes 100% of that makes it out to the Gulf of Mexico. Farther up, fertilizer that's applied to fields that are farther away from the outlet, even though that is enters the streams, those streams, as they're traveling from down the length of the Missouri, the Mississippi, as they're reaching down towards the mouth of the Mississippi, they are losing nitrogen. That nitrogen is being processed by in-stream microbes. It's occurring both in the channel and, importantly, it's occurring in the bed and banks of the channel, in the groundwater associated with it, and it's occurring in floodplains and off-channel wetland areas. This is not only the Mississippi. If we look closer to home here, this is the Ipswich River watershed. This is flowing from south to north, west to east in northeastern Massachusetts, entering Plum Island Estuary. You can see that concentration measurements were taken down the length, down the main stem of this river channel. And if we just look at how those should be diluted by relatively clean groundwater, we would expect that the concentration would follow that red line. But instead, measurements have shown that there's a reduction in the dissolved inorganic nitrogen that's delivered to the mouth of the Ipswich. And wetlands in the upstream portion of this watershed are playing a really important role in this process. When we're looking at river networks, it's not only the average that matters. We're getting loading of nutrients and pollutants into our rivers. They're vecting downstream. They're dispersing and spreading out in the main channel. But they're also interacting with transient storage zones, both in the channel, side pools, step pools behind logs and boulders, in wetlands that are off to the side, and also in the hyporheic zone, the region of shallow groundwater under and next to the river. Those are regions where we tend to have enhanced levels of biogeochemical processing due to the redox conditions that are present there and the microbial communities that they support. So if we're looking at a reach scale, if we're trying to understand what a river is delivering to downstream water bodies, we need to take several things into account. we need to take into account the loading, how much of the nutrient or the pollutant is entering, and where is that occurring? If it's occurring very close to the exit, there may just not be enough time within the channel for processing to occur. And if loading is very low, then uptake may be low because there just may not be very much around to process. Biogeochemistry is also extremely important. If we don't have favorable conditions for uptake, then there just may not be a lot of reactivity present. But in addition to having those sites for biogeochemical processing, we also need to make sure that the dissolved solutes come into contact with those areas. And so there needs to be connectivity and a long detention time so that the solutes can interact with those areas of reactivity and uptake can occur. So we can see source limitation, or that uptake may be limited by the source, by reactivity, or by transport. I'm a physical scientist, and I am most interested in the physical, or I'm going to talk today about the transport limitation. So an area in which we have enough solute available. There are some biogeochemically favorable zones for uptake within the reach, and we're interested in the transport access of those. So one way, if we're looking at reach scale uptake, we're really interested in the change of mass flux from the upstream to the downstream end. We're interested in how much, whether that is reduced or whether it increases as we're moving downstream. However, just looking at that sort of black box approach, looking at those reach scale averages, it's affected by all three of these aspects. And so we cannot disentangle those effects just by looking at a solute mass balance. So if we want to understand what's driving that solute mass balance, that overall reach scale uptake, then we need to use some different tools. And one of the tools that we can use is to use a introduced tracer. So a tracer, dissolved tracer, is a substance that we add. Hopefully it's non-toxic. Hopefully it's present in fairly low concentrations so that we can easily measure it down to low concentrations. And hopefully it's conservative, so it is not reactive. And therefore we can measure how that dissolved substance that we've added in known quantities moves. And when we look at that, we therefore know how other substances that may be reactive, that may be more toxic, that we may actually care about. Typically, in a river system, we release the solute upstream. Here, this is showing a slug or a pulse release where when it is all introduced at once, and it then moves downstream. Some distance downstream, it mixes across the width of the river. Downstream of that location, the details of how we injected it no longer matter. And we typically measure it downstream of that location at some fixed sampling site here. If we sample generally as that cloud passes us, we see that the concentration increases and then decreases, and there may be a very long tail due to the interaction and sort of the fact that some of those solute molecules get stuck in longer flow paths. And importantly, if we understand how this conservative tracer moves, then we can understand how reactive substances move, and therefore we can sort of disentangle, we can deconvolve the reactivity from their transport. We analyze these studies usually using a breakthrough curve. So this is a plot of the background corrected concentration, the excess concentration at a stationary location downstream. And one of the things that we can get out of this is the recovered mass. We can see how much of that release tracer we observe. We can just do that by integrating the breakthrough curve and multiplied by the discharge that is measured at the downstream end. Both of these may be time varying, although it's easier if the discharge is steady. On the right, you can see, well, both of these are, on the slide, you can see two measured breakthrough curves. The one on the right is plateau, reaches a plateau concentration, a steady concentration, because the solute was released continuously over a long period of time. If we have a slug release, or sometimes called a pulse release when everything entered at once, this produces a characteristic curve where there's a rising limb up to a peak, then a falling limb, and then potentially a long tail. And we typically interpret this slug release breakthrough curve, which It represents the residence time distribution of water parcels marked by our introduced conservative tracer. It represents how long they're spending within the reach. We can use the peak of that curve to give us an idea about advection by the mean flow within the stream. We can use the width of that curve to give us an idea of in-channel dispersion or spreading. And then the tail gives us some information about transient storage. I'm going to be a little more specific about exactly how we might interpret this and give you some, canonical curves that we might observe. So first, the simplest breakthrough curve shape that we might see is what we would see if we have steady flow through a uniform channel with no transient storage. The advection, the governing equation in a 1D system where it is mixed across the cross-section is just the 1D advection dispersion equation, where the concentration at time at our measurement location changes over time in response to advection and in-channel dispersion, which we typically assumed to follow Fick's second law. This dispersion coefficient bundles together molecular diffusion, but also the influence of turbulence, shear dispersion, dead zone dispersion, any other spreading process that's present within the main channel. And the solution to this equation, I'm sure most of you know it, is a Gaussian curve. So this gives us it's written right here, M is the mass released, A is the cross-sectional area of the stream, and then we get this equation right there. If we non-dimensionalize it, we can introduce these three, or we can rewrite it in terms of these three terms here. C star is the fully mixed concentration, which is a theoretical quantity that we would get if we dumped all of the tracer in and it instantly mixed within and only within our study reach. Very theoretical because really we have a pulse that's moving downstream, but at least it gives us a scale for the concentration. Tau is then the advection timescale, the length traveled divided by the mean flow speed, and the Peckley number represents the ratio between the timescale for advection and the timescale for spreading. My student Jia Yang sort of plotted some curves that I find helpful in sort of thinking about these equations, so I might as well share them with you. Here you can see the influence of the C star parameter, this theoretical fully mixed concentration, and not surprisingly, if you increase the fully mixed concentration, this here is increasing by a factor of 100, the concentration that's measured anywhere within the reach increases. If you increase the center of mass arrival time, then you arrive later. Makes sense. Curve is shifted laterally. And if you increase the Peckley number, the influence of spreading, the curve is much wider and the peak concentration is reduced to preserve mass within the curve. Okay, so that's the simplest situation. What happens if we have transient storage? Let's first start by just considering a single transient storage zone. The governing equation here is modified to account for mass transfer between the main channel and the transient storage zones. We get separate governing equations for the main channel and the transient storage zone, which is typified by a single time varying concentration. And the solution to this, one solution to this was proposed by de Smet. It's based on the Bessel functions, and you can see the Gaussian solution right here, and then that is modified with this additional term that gives us a longer tail. So there are two parameters. There's this alpha parameter and this beta parameter. Alpha is the mass transfer coefficient between the mainstream channel and the transient storage zone. Beta is the size of the transient storage zone. And here are some figures that show how changes in the mass exchange rate and that aspect ratio affect the curves. And you can see that there's sort of a trade-off between how much you have in the peak and how much you have in the tail. As you increase transfer to the transient storage zone, the tail gets bigger and the peak gets smaller. One thing that we often see in the field is that we don't just have a single transient storage zone typified by a single residence time. Instead, we have multiple transient storage zones. We have some with long residence times and some with short. And that gives us a very long tail, which sort of shows us the influence of all of those transient storage zones convolved together. One way of representing this is a single uniform hyporheic zone beneath the steam channel, where the material right at the top exchanges very quickly with the surface water, and farther down it exchanges more slowly, but it's uniform over the length of the reach. And if we make that assumption, this is the governing equation that we get. You can see that we're now modifying our 1D transport equation by adding in this exchange term, which depends on the vertical hyporheic concentration gradient at the stream bed, as well as a homogeneous steady vertical diffusion coefficient in the hyporheic zone. The solution to this equation is shown here. Lambda is a dummy variable. Again, this depends on the Gaussian solution, and then it's modified by these additional terms, which include this mass exchange coefficient alpha star between the main channel and the hyporheic zone. And that alpha star parameter, see, this is what it does. Again, it sort of reflects the balance between the peak and the tail of the curve. So, armed with that, I'm going to give you some questions that I'm going to talk about today. I am optimistically going to try to give you a quick view of 4 projects, student projects that I've worked on. We'll see if I get through that. I'm being optimistic. And through these projects, I'm going to try to get to questions such as this. How does transient storage affect the transport of dissolved solutes through river networks? What factors affect the amount and connectivity of transient storage? And what is the expected impact on reach scale uptake? Question?
What is the typical tracer size that you use in these experiments?
Typical tracer size, what do you mean?
The
size of the molecule?
Yes.
Different studies use different, typical tracers are salt, so just sodium chloride, or rhodamine WT, which is a fluorescent tracer, is also used. We're hoping that all of these are dissolved, and so the molecular weight shouldn't really matter because it's really turbulence, it's differential advection that's affecting any dissolved solute. So any dissolved solute will move similarly to any other dissolved molecule. At least that's our hope.
And when you said that the diffusion coefficient actually takes into account some of the other physical phenomena at play also.
Yes.
By what factor does it really change if you were to just do not in a flow, but if you were to just diffuse in like a glass, if you were to put that and measure the diffusion constant versus the effect of all the other physical phenomena, how much does that affect the diffusion constant?
The diffusion or dispersion coefficients that we're measuring in the field, they're apparent, so they come out of the flow, and you can't measure them in a lab glass because you don't have the process, you don't have the turbulence, you don't have the shear, you don't have all of those processes present.
You can measure a baseline.
So you could measure a molecular diffusion coefficient by looking at it in a microscope, but that is going to be dwarfed. It's going to be overwhelmed by the fact that there is shear within a river, that the top of a river is moving much faster than the bottom of the river. That is going to rapidly spread out a tracer cloud, way faster than Brownian motion. So yeah, this is sort of an emergent coefficient that is measured at the same scale as sort of it's a reach scale spreading coefficient, which yes, has a lot of hand waving. I totally admit that, and
as long as it works, I mean.
Right. So the idea is that we want to measure sort of, well, what's there? Okay. So I'm going to start by taking us to the Suncook River in Epsom, which is just a little bit west on Route 4. If you've driven to Concord, you've driven you've driven here, right through the Epsom traffic circle, and just south of there, you may or may not be aware, the river, there was a major avulsion in 2006. In a very large flood, the river jumped out of its former river channels and it carved a totally new river channel through a glacial deposit. Our study had nothing to do with that, but it did spur the USGS to fly some very high resolution green LiDAR of this whole reach. This was the Earl B system. The average point spacing was 0.52. This was flown in 2013. And if we zoom in on a couple of these areas, you can see some of the detail that is present within this data set. So this is giving us not only the floodplain overbank areas, which is what we normally can get with LiDAR, but it's also giving us the bathymetry, the bed of the river underneath. And it's picking up really detailed bars and features on the bed, which create roughness, which are affecting the flow. So this was a fantastic data set, and we therefore tried to see how is the new channel contributing to solute transport and in particular nitrate retention. My student Megan went out and did some rhodamine releases. Here is Megan releasing some rhodamine. Maybe color isn't coming through very well, but you can see the sort of red color right there, flows from right to left. We had a video camera station downstream. 11 minutes later, we couldn't really see the rhodamine yet. By 13 minutes, you can maybe see that the main channel of the stream is starting to turn a little pink. At 14 minutes, not only was the main channel pink, but also this sort of off-channel little side area. It took a little bit longer to get there, but it eventually turned pink. By 17 minutes, the main flow of the river was no longer pink, but it was still present in this side area. And then back by 23 minutes, we can no longer visually see it within at this sampling location. Using our eyes is maybe not the most quantitative measurement, so we also had a fluorometer in the stream that was measuring the concentration, and you can see this black line. This is showing what that fluorometer is measuring. And so we see this rapid rise to a peak, and then a long tail, much longer than we would expect for a Gaussian curve. So Megan calculated this Gaussian curve sort of using the first half of that peak, using the rising limb, what it would, if it were just in-channel dispersion that were contributing to longitudinal spreading, then the concentration should return to baseline relatively quickly. But instead, we see this long peak. The green line is showing discharge. The discharge axis is on the right. The green line is showing discharge from a rating curve and a pressure transducer. And it was relatively constant. The green diamonds are showing us the discharge that was actually measured at the location, which is close to the rating curve in some cases, but in some cases, maybe our rating curve isn't the most accurate. But regardless, we're getting experiments during very different discharges. Our lowest discharge, the discharge was a little bit less than 0.6 cubic meters per second. Our highest was at or nearly over 15 cubic meters per second. So a wide range of discharges through this reach. Oh, I should mention, from these, Megan extracted the time of the peak arrival, which is shown as that dot dash line, and then the detention time, the first moment of the distribution, which is shown as the dashed line. In all cases, the detention time, the first moment was after the peak arrival because of that long tail. And from those arrival times and knowing our reach length, we can get a transport velocity, which is shown here. And that transport velocity changed as a function of discharge. In general, we see, if you squint, an increase in both the peak arrival time and the mean time, the mean velocity calculated from the detention time, sort of at all of these rivers. The blue squares are showing another study done by Smith et al., showing peak velocity for a wide range of New Hampshire rivers, and we're getting velocities that are similar to other rivers. And if you sort of look at multiple rivers, we see the same effect. In general, as you increase discharge, velocity increases. Similarly, if you increase discharge, dispersion increases as well. So this is the width of the curve related to the width of the peak, and higher discharge leads to a higher dispersion. We were finding dispersion coefficients that were a little bit higher than the older study done by Smith, probably because he was using graph samples, and so he couldn't really get the he wasn't as worried about capturing the full width or the full tail. So he may have underestimated the dispersion coefficient for his streams. So we have our tracer studies. We also took field velocity measurements at each of those discharges. We have very good bathymetry. We had some estimates of roughness. I'm skipping the details, but we put this into a hydraulic model, Delft 3D. And from that, we were able to represent the flow field. This was a 2D model, so not only, so it was resolved over the width and the depth. This did a pretty good job simulating the arrival times sort of for our tracer releases. And here on the right, you can see a map of what this model, this is just an inundation map. As we're increasing discharge, we're increasing the area of the riverbed that is inundated by the flow. With this hydraulic model, we were then able to continuously load a reactive solute chosen to represent nitrate. And this is what we get. First, before we can look at what we get, I should say, we have to make an assumption about where nitrate is being taken up within the stream. And we didn't have very good handle on that. And so we tried 3 different simulations with 3 different assumptions about what could be going on. Scenario 1, we had a spatially uniform first order uptake rate constant. And we have high concentrations entering the reach and then low concentrations going out. That makes sense. For our second condition, we have spatially uniform uptake velocity. So this is what happens if you have all of the uptake occur on the bed of the stream, then you have a constant uptake velocity. And that similarly, we have high concentrations entering, low concentrations exiting. But you can start to see that there's some regions that have very low concentration, well, comparatively low concentration, which are really shallow areas. And so therefore, any solute that reaches that has more contact time with the bed. And so the concentration may drop more in those areas. And then our third scenario was an uptake velocity that was chosen. We had 2 different uptake velocities that were possible. One that we applied where the shear stress was high, and then a higher one, a boost in uptake that we applied when the shear stress was low. And that gives us this simulation over here, which has a slightly different spatial distribution of where the uptake is occurring. But all of these models, when we look at them, give us that the fraction of nitrate retained as the discharge increases drops off. There is, you can't really see, but there's actually for very low discharges, our highest retention was the second lowest discharge that we compared. We think because as we increase the discharge, the model was able to access more, or the water was able to access more of the bed. And as it was able to access more of the bed, it was therefore able to reach more of the bed area, which is a site of a lot of biogeochemical uptake in these systems. So to summarize this study, we used a numerical model to simulate temporal changes in reach scale reactive solute uptake. In general, retention increased at lower discharge, and we can use the curves that we came up with to predict what discharge or what nitrate retention, how it might have varied temporally over the course of the summer. And so during July, where we had a very high flow, we would have expected very low retention. But then later on in the summer, as we got summer low flows, retention rates were expected to have increased. Holding everything else constant, holding loading constant and holding biogeochemistry constant, which does not make sense here exactly because we know that there's seasonality in those reaction rates. Interestingly, the reach scale removal rates that we got were different than the ones that we put into our model for the individual patch or grid scale, because the reach scale rates took into account not only those local rates, but also the connectivity of the flow to those environments. And this study did not consider those biogeochemical influences, additional nitrogen loading, or exchange with the groundwater. To talk about exchange with the groundwater, I have a study for you. This is one that is currently going on. I have 2 master's students working on this up in the Zealand River in northern New Hampshire. The Zealand River is right on the west side of Mount Washington. It drains, well, I guess the Ammonoosuc drains Mount Washington, and the Zealand River flows from south to north into the Ammonoosuc, upstream of the USGS gauge. This is the LiDAR hillshade for the area. You can see it's a valley carved by the glaciers. And again, the Zealand River is flowing through this valley. There's a long legacy of human modifications here. Back in the 1800s, the entire area was clear cut, and the steam-powered locomotives created many forest fires. There was a lot of sediment that got into the stream channels, and they were very unstable. To try to deal with very unstable channels, in the 1960s, they went in and put in these gabions. They channelized the river. It started to incise very deeply and started to.
What's a gabion?
Gabion are they're cages of rock.
Okay.
If you had big rocks, that would resist flow. That would resist the flow. However, if you don't have enough big rocks, you can take small rocks and put them in a cage. The problem with a cage is that it eventually rusts, and then you have a very dangerous failing cage that whips around like a snake during flood. So there are some problems with this system. And as a result, in 2024, the Forest Service worried about the fact that the river was moving towards a forest road and a water supply pipeline for a local town. They reactivated an old channel. They went in with a lot of heavy equipment, and they cut a new channel out onto the floodplain. And then they rerouted the flow so that it now went onto the floodplain and no longer in the old channel of this river. Again, I'm not going to talk to you about this portion of the study, sort of evaluating the restoration, but it's interesting. So here's a map. This is showing the LiDAR of the Zealand, where the Zealand Road is. This is the old channel, and you can see that the old channel was very close to the road. And so they went in at this diversion site and they carved a new channel and then just allowed it to flow out onto the floodplain, taking advantage of very old relic channels that were present. The old channel is now only fed by a little tiny brook, Hale Brook, which is coming off of the mountains. An aspect of this study is to try to estimate channel roughness, and I know that many of you are interested in mapping. I have a student who is generating these beautiful pictures of grain size throughout this reach here. This is showing the D84. Red is bigger, green is smaller, using a drone, point returns from that drone, and then those point clouds to look at the roughness within various kernels to try to get an idea of grain size. If you are interested in this, look out for Ryan's thesis defense, which is coming up in a few weeks, and he would love to tell you more. He's the expert on that. I'm instead going to talk to you about what happened with groundwater during this system. During the summer of 2025, and if you remember last summer, there was a drought. And so this is the Ammonoosuc discharge. The Ammonoosuc started fairly normal for the year, but then later on, the Ammonoosuc was starting to set new low flow records as a result of the drought. If we compare that to what we're measuring within our reach, we're measuring here are the logger locations. Coming in, we have yellow, and that tends to be fairly high. And the channel is losing, the blue line is lower than the yellow line because not all of the water is staying in the channel. We tried to prove this using salt. Again, we went out, we dumped in some dissolved salt, we measured the concentration downstream, and just to show you what the recovery is, we recovered 67% of the mass that was released, which means that 33% of the mass was lost, and therefore 33% of the flow within the water, within the river that was marked by the salt was also lost. We can compare that to what we get from a groundwater model of the system. The total lateral flux that we predict from that model was 0.013, which is a little bit less than we observed stream losing. So this is trying to show that here. What we measured with the salt release was shown as that blue dot. The green dot is showing us what we got for groundwater, and that's a little bit less, but it makes sense that the stream might have been losing both to the left and to the right, whereas with our mod flow model, we were only picking up flow that was going to the left in one direction. And then the black line here is showing the measured difference in discharge between the upstream and downstream locations. And in general, we're getting negative numbers during the drought, indicating that the channel was losing water and that it was losing more and more and more as the summer drought kept going. So the fact that we have multiple channels is spurring groundwater flow through this area. It is increasing exchange with the hyporheic zone, and we didn't look directly at solute transport or uptake, but if there were solutes that were taken up in the subsurface, suspended sediment, nitrate, we would have expected that losses would have increased as a result of this. Look for upcoming thesis defense from Rupert Coriati in the Masters of Water Resources Engineering program if you're interested in learning more about this. This is River's project, which they kindly allowed me to give you a teaser on today. In addition to, when we're looking at streams, another very important aspect is temperature. And so when we're looking at groundwater inflows, we can actually pick that up with a thermal imagery. The black here is showing very cold water that's flowing into a puddle here. This is sort of in the diversion site. You can see that our stream water is pretty cold. And when it's flowing through the ground and then coming out into a warm puddle that's in the sun, we can see that groundwater coming in. Again, sort of underscoring this influence of groundwater exchange between groundwater and surface water here. So we're trying to understand here, sort of adding in this aspect of groundwater, natural river channels do exchange water with the ground, and those fluxes vary seasonally and in response to flow conditions. I had initially planned to talk to you about some work within wetlands. Maybe I'll go through this extremely quickly. So these are 4 watersheds nearby. And in each of these, again, we used a conservative tracer to try to understand how solutes were moving through them. And what we see here is that multiple surface transient storage zones were present. This picture is just showing, we're getting fast flow in the wetland channel. You can see the diacloud moving past. It started to move a little bit into nearby areas, but then these longer flow paths, it hasn't yet permeated into these areas of the wetland that are a little farther away. And when we look at the breakthrough curve of rhodamine at the outlet, we see that a large portion of the tracer is not traveling within the channel. Instead, it's in this long tail. Sophie used a multiple storage zone representation model to try to understand how this was moving. I will skip the details. She was able to represent the breakthrough curve fairly well. And the output from her model was that surface transient storage, or the fraction of time that solute molecules spent in transient storage within these wetland-dominated reaches is very high, much larger than we would expect for streams. So which makes sense. So we're getting residence times that are 60–70% of the time that it takes those molecules to travel is because they're interacting with these off-channel areas. The one exception to that was a study that we did during a drought, which was so dry that it was not able to access the off-channel areas. And so it really was behaving more like a channel, with like a stream channel than a wetland channel. And in addition, not only did the mean residence time increase, but also the variance of the distribution increased. Streams exhibit behavior that is above what we would expect from Fickian transport. So this is sort of a historical study of streams. And if we add wetlands, we're getting even more transient storage or even more variance in the curve than we would expect from streams. So the conclusion from this is that in-channel wetlands substantially increase residence time, retention time, and therefore the opportunity for nutrient processing. And that's not even considering the fact that wetlands often contain very active biogeochemical communities where reaction rates are actually higher. The last study that I want to talk about is a synthesis study that my student Jiaying Liu defended last year. Jiaying is a mathematician, and so he did not go and perform any tracer releases, but he contacted a bunch of scientists and looked at a bunch of data banks and collected. He formed a data set that took into or included tracer releases from 29 rivers of varying sizes from across. Most of them were from the continental US. There was a few from Alaska and then a few from Northern Europe. And he tried fitting these breakthrough curves with a curve that took into account not only the peak, which he used a Gaussian for, and the near portion of the tail, but he also tried to fit this longer portion of the tail with a power law distribution, creating a combined fit that included multiple parameters. And different field breakthrough curves exhibited different shapes. One of the big impacts on this is probably due to truncation. If you cut off the tails, if you stop measuring before you're all the way back to background, maybe because your instruments are not very sensitive, or there's a background concentration present in the stream, or it's time to go to bed and you're ready to go home, then it's difficult to resolve the full length of the tails. The coefficients that he used, I introduced these to you early in the talk. These are the same ones. The reach average concentration, the Peckley number, the advection timescale, the hyporheic mass exchange coefficient, and then the slopes of the tails. There were some differences between these different streams. You can see that these boxes, there's some variation from river to river to river. The type of the curve didn't really matter that much, suggesting that, although we can't resolve these far tail slopes if the curve does not continue long enough. When we compared how well the curve did at actually fitting the breakthrough curve, it did a very good job. Very high when you go to efficiency, close to 1. And so we concluded that this accurately captures the slug release breakthrough curve, and therefore can be used to improve planning, improve interpolation or extrapolation, and allow us to compare solute transport between streams, which was really our purpose here. And the 8 fitting parameters that we have were nominally related to measurable characteristics. So for example, we can, from the metadata that goes with the stream, if we know how long the reach was and what scientists at the time measured the velocity in the stream to be, we can get a pretty good idea of what it looks like solute, if we sort of fit a curve and see how fast the solute was actually behaving. If you squint, there is a positive relationship there. It is not a perfect relationship. And that is in part because when you go and measure the velocity within a stream, you're measuring it generally at one cross section. And what we're getting from these solute studies is really an average over the entire reach, and that may be different. We know that streams flow faster in some regions and slower in other regions. Full disclosure, this was the only really strong relationship that we saw. The other stuff, it's just very hard to measure these things in the field, or not all scientists report all of these parameters. Not all of them report how much tracer they released, those kinds of things. And so future studies will have to try to do a better job of collecting some of that metadata, as well as getting scientists to post their data in repositories. We tried to simulate this using a particle tracking model. I will skip the details. We did a pretty good job, and then tried to also estimate them using random forest to train a model to predict these parameters based on knowing something about the stream input. And I think I'm going, and to summarize, if we gave these machine learning models a lot of information about the stream, so if we gave it some information about the stream surface flow and the subsurface flow conditions, then we did a really good job predicting the tail of this distribution. It was terrific. These are the results. I know there's some scattered, but just trust me, I'm about to show you some really bad data, because most of those the tail prediction really depended on this subsurface transport, which is really hard to measure in the field. And so when we dialed back the information that we gave our models to only stuff that we really could measure easily, the reach length, the reach depth, the slope of the stream, and the surface velocity, and then we tried to predict the entire curve, we did a really good job with the average flow velocity. We did a decent job with the Peckley number, but everything else is just noise or scatter. And so our takeaway from this is that if you're trying to understand these long flow paths, which is really where a lot of biogeochemical processing is occurring, you need to know something about the transient storage. You need to know something about, for your stream, how much time does the flow spend in these slow flowing biogeochemically active areas. And that is hard to measure. That's one of the reasons that Solute releases, although it's not a new technology, they're still really used. They're still used frequently. When we applied this to our field breakthrough curves, we did an even more horrible job. However, our reach scale solute uptake prediction was still relatively strong. So reach scale uptake was relatively robust, even though we were missing the details of the breakthrough curve. And that's really because a lot of the uptake occurs not in the very ends of the distribution, the very longest residence times. It really occurs in the sort of the part just after the peak, where solute molecules have come into contact with very accessible transient storage. The very long flowing transient storage, not as many molecules go into them. They're not as connected, and so they're not as important. Okay, so back to our research questions. I can put these up. I think that we learned something about transient storage, or at least I talked about it a lot. Tried to convince you that transient storage is important in understanding the transport of dissolved solutes. It depends on discharge. It may change seasonally and with floods events. And if we're looking at reach scale uptake, readily accessible transient storage has the largest net impact on reach scale removal and effective reach scale uptake. That's what we see at the reach scale, scaled up to that reach, depends both on the uptake rate within hotspots and also how well connected they are. So just to put this back on the coastal ocean, here's a picture of the Merrimack, which I did not study at all, but it is a mouth of a river. And so I'm just having you imagine that this flow is going out into the ocean. And some overall conclusions, transient storage within rivers is important, I think, even if you are an ocean person. And yeah, here are some conclusions. Thank you very much.
Questions?
And very nice, and thanks for giving the presentation today. So I'm curious about the modeling that you did.
Yeah.
Oh. And how do you estimate what that bottom drag coefficient might be for that model? And how important is that, considering you have slope and bottom is going to drag the flow?
We did not do a sensitivity analysis to it. We assumed it's a sand bed river, and so we just assumed sieved 1 or 2 samples, got a median grain size, and then just applied it spatially. One of the reasons that Ryan is really trying to do grain size for the Zealand, which is a cobble bed river, is that varies, and there's a huge impact on the hydraulics, depending on where it's more gravelly, there's no sand in there, but where it's a bigger cobble boulder or a smaller gravel.
So you try to do spatially varying.
So he's doing spatially varying drag. He's doing that. The overbank areas, he's using the LiDAR. The in-bank, we're just doing sort of couple counts and then visual sectioning of that.
But you try to at least look at what's there to give you some idea what that number would be.
Yeah, he's not planning on doing sensitivity, although you're certainly welcome to encourage him to extend his thesis to do that. But he is trying to do his best at capturing that spatial variability in the roughness. Jamie?
So you have these transient slugs.
Yes.
Are there longer time storage reservoirs which they'll miss? Like if you put a 20-minute pulse in, could it be missing the effects of reservoirs that take an hour to go into, an hour to get out of? And are those important?
I have two answers to that. One, if you wait long enough, you'll see most of it. Although you're waiting long enough means that you need to have a tracer with a very low detection limit because you're really measuring very low concentrations in the tail. And even if you do wait long enough, it's possible that there's some very deep hyporheic flow path that completely bypasses your sampling probe. But certainly, better detection limits can help with that problem. However, when we look at the impact on, this all comes down to where the reactive zones are. Reactive zones are often right at the bed of the stream or just below it, because that's where you have the interface between the oxygenated water and the anoxic groundwater. And so that's really a region of very high reactivity. And so what we see is really the deep flow paths, they're not as important. And so we're not as worried. So Jiaying has done all this work trying to capture, the very end of the tail. But then when he looks at, does it matter? Not so much. I think just taking 6 grab samples to get the peak is probably not enough, but waiting days seems like overkill for most of these systems, depending on where the reactive sites are located.
Like imagine you had a pollutant that was going down there for days and days. Would that make the tail, once you cease the pollution, last much longer? Because it had
so theoretically, if you integrate the slug release, you get the cumulative distribution, which is the breakthrough of a continuous release or a plateau addition. If you're really interested in the tails, those plateau additions are used more frequently because they do a better job of sort of capturing that very long tail. I guess with a pollutant, so the whole idea of using solute, using a tracer to represent another substance is that we can, they behave similarly. I guess with your example of a pollutant, maybe at some point you're bringing up this idea that it saturates and, you can no longer remove any more. And so that issue of saturation of the reactive zones is sort of separate from this. This is assuming that the reactivity continues at the same rate, which, depending on the chemistry of what you're looking at, may be a terrible assumption. Well, you have a question.
In the very beginning of the talk, you showed a lovely meandering stream with an input where you dump the slug and an outlet at the other end.
The Wind River.
And it was doing this. Longitudinal distance to me is the straight line between beginning and end, and not the actual flow direction or distance that the water has flowed. Which is that variable? Is it the longitudinal distance from A to B, straight line?
So I guess
actual
as
distance.
we're thinking about distance, it would be the meandering distance. So it would take into account the straight line times the sinuosity of the river.
Okay, so
you're right that if other people are reporting it differently, that maybe is why we're getting some additional scatter in our curves.
Okay, it would be useful to point out that you are measuring the actual limit.
Yes.
Question 2 is you've got something called water velocity. Where is that measured? And would it make a big difference in your model if I took you to a youthful stream, which had a very high gradient, versus the streams that you've been looking at that have typically very low gradients?
So Jiaying, his synthesis study included both high gradient and low gradient streams, but that velocity, that is a reach average velocity. So it's not on the left, it's not on the right, it's not on the upstream side, it's not at the downstream side, it's a magic average over all of that variability.
We all use magic, don't we?
Exactly.
Thank you.
So we have one from Catherine Alder. Really interesting work. I remember dispersion coefficient models from 10 years ago were about 5.9 times friction velocity times depth. Is your work related to advancing those models for practitioners who don't have resources for numerical modeling?
So, so one of the hopes of this line of work is to come up with those rules of thumb to try to allow people to plan for understanding how flow is going within rivers. I showed at some point sort of a comparison of this study to more classic river studies. And when you add wetlands, you tend to increase the amount of dispersion above what you would expect just from the log layer flow, which is what that particular estimation is based on. Cool.
I wanna model some transport between river and estuary.
Yeah.
What kind of dynamics may need to be considered additionally?
I feel like you're the expert on that.
I just wonder, yeah. I want my colleague wanna model them, but I'm near the ocean side. My colleague is in some show, he get a degree here, in the hydrology. We have no knowledge between those, between our sides. So I just wonder about your idea here.
This would sort of be subgrid scale mixing, that if you're doing a model of an area, you parameterize that subgrid, those subgrid scale processes, right? And you have to elevate the effective dispersion coefficient to account for that. And so maybe that would be comparable.
Yeah.
I guess another, this idea of flow going into the bed and having a lot of reactivity. I know that there have been some studies in coastal areas that it's very important in the coastal area as well to sort of have that subsurface access, under bed forms or ripples. So that might be comparable. So definitely similar processes, but not identical.
Thank you so much. Yeah.
This might be a very naive question. Since I'm not from the field, I will ask it anyway. How is the tracer concentration actually measured in the field? What is there in this particular spatial arrangement in which you
Sure, it depends on the tracer that is being measured. For rhodamine, we use a fluorometer, which I'm forgetting exactly, but rhodamine absorbs at it absorbs at 532 and it releases at, I don't know, nanometers at something like that. So you measure the fluorescence coming off of the molecules after they're excited by a laser beam at a particular elevation or a particular frequency. You have to calibrate the voltage response to rhodamine, and then you also there's an interaction with dissolved organic matter, chlorophyll, within the area, so you often have to do a field calibration as well. But yeah, you're looking at the voltage from the fluorescence response and calibrating that to a concentration. For salt, we often use conductivity, which again you need to calibrate. So we're often doing a derived metric and then calibrating that to get a concentration off of it.
And that gives you information across the cross section where you measure it.
So the idea is that you're measuring downstream of where it's mixed across the cross section. So it doesn't matter whether you're measuring on the left or the right. They should be giving the same breakthrough, the same concentration. In the field, we often try to take measurements at the left and the right and verify that they're the same. But hopefully we've placed our probe down enough that yes, they match. And we can assume it's a one-dimensional system. Yeah.
That was a great presentation. I have a question on the Zealand River thermal imagery. How does getting the surface temperature tell you about the groundwater storage if the top is warmed? Why would it mix?
So during the summer, groundwater is cold. Groundwater is at the annual average air temperature, so it's colder than the surface water. And you can only detect it when you have a plume of groundwater coming in at the surface. Then you can see that it is cold from the thermal imagery. If it's coming in the subsurface, or at the bed, you can't see it on the surface usually because it's denser and so it doesn't rise up. So you would need to use a distributed temperature sensor or something else in order to pick up that cold water input.
We'll look for the brook trout that seems to sit there.
You could just look for the brookies, exactly.
No questions? I guess then we can thank